So what we're going to talk about today is what you're probably in this class for. What are the microbes that live on the human body, on and in the human body? These days we call this the microbiome. It's what people used to call the normal flora. Surprisingly, we don't have very good answers to questions about the human microbiome. This is where microbiology started, right? What causes diseases and where are the organisms there? But what do we don't know? We don't know how much diversity there is in and on the human body. Even fundamental questions. Is amniotic fluid sterile? Is even the bloodstream serum sterile? Probably, but we're not sure. How much do different microbial communities differ on different parts of your body? The fact that they do differ is obvious. The microbiome of your gut is very different than the microbiome of the fluid in your eyes or in your ears or whatever. But how do they differ? How much are they different? How much variation is there in normal microbial communities? If I take a swab from my hands, is it going to be the same as yours or yours or yours? How are they going to be different? How are they going to be the same? How are they going to be different? We really don't have very good answers to that. How much do they change over time? So there's some information on that in terms of things like the oral microbiome. It's clear that when teeth come up, they go through a normal ecological succession in terms of the biofilms that grow on the surface of the teeth. But through your lifespan, how does your microbiome change? The answer is nobody knows. And this is a big one. How do those microbiomes contribute to your overall health? If you don't get anything out of this lecture at all, other than this one point, please take this one point home. Stop using antimicrobial creams on your hands. All right? Those things are terrible for a variety of reasons. One of them is most of them contain ethanol. It dries out your hands, you create cracks in your skin, and it makes the problem worse rather than better. Unless you've got a specific reason to, if you've got the flu or if you think you might have the flu, then yeah, okay, those things are pretty good at killing flu viruses and things. But in most circumstances, those things are terrible. We live in a world these days where culture will tell you that everything has got to be clean and pristine, that sterile is good. Kids' toys have antimicrobials soaked into them. This is such a terrible thing. This is what we look like. This is an old-growth tree. My view is that if you want to assess the health of this creature, just looking at the oak part of it is a mistake. Because this is a community, right? It's got vines, it's got parasitic plants, it's got things down here in the roots, it's got bacteria, it's got archaea, it's got insects. This is a giant community. And the oak part of this cannot be healthy unless that whole community is healthy. Any of you guys keep aquariums or know someone who does? So the rule in aquariums is there's no such thing as a sick fish. There's sick water, but not sick fish. If the water is good, then the fish are going to be healthy. And the same is true for other kinds of environments, like this one. If you're interested in this organism, and you want to know the genetic capabilities, you can't just sequence the oak genome. You've got to know what else is there and how that contributes toward the function of that. Now, I understand that you don't like to think of yourself this way, right? But there really are mites that live in the pores of your skin, and they don't do you any harm. There's no reason to be grossed out by that. One of the real problems that this generates is when you look at all the organisms in your body, what is normal? There are specific situations where it's easy to tell that something is not normal. If you've got tuberculosis, you've got a particular organism growing in your lungs. And that's easy to identify. And most of microbiology is those kinds of situations. So you've got a specific disease caused by a specific organism. Strep throat, syphilis, whatever. Many microbial diseases do not fall into these categories. Crohn's disease, prostatitis, and the list goes on and on. Those are a lot harder. Those are diseases caused by problems with your microbial community. Gingivitis. What's the causative agent of gingivitis? There's no answer, right? Because it's a disruption of the normal flora. But in order to identify those kind of situations, you have to know what is good, right? And that's a problem. So the paper we're going to talk today about is from Rob Knight's lab, and it's an attempt to answer, or at least in part, or begin to answer that very question. So what have they done? The purpose is to identify human-associated microbial communities, how they vary from place to place on the body, from person to person, and within a single person over time. So they've taken a bunch of samples, 4 different days, 1 day apart, and 3 months apart. Yeah, 3 months apart. From all of these body locations, and they've got 9 people. Yeah.
Is that span of time sufficient
To begin with, I mean, your question is, of course, perfectly valid. It would be really interesting to go through a person throughout their lifespan. But this was within the constraints of what they could do at the time. And it's a start. It would be nice to do it 1 full year later. I think that would have been an improvement. And so things like the forehead, the external nose, the right ear, the left ear, armpit, left and right. Notice they're doing left and right a lot. Any idea how you're bilaterally symmetrical, right, at least on the outside? How do those differ? Yeah, so particularly your hands, right? And so there's going to be some differences there that are really interesting. And so forth and so on. Notice that these are swabs. And so from my perspective, the biggest caveat to this experiment is that you're only looking, well, number one, you're only looking at ratios of organisms. You're not looking at the full quantity. The way the experiment is set up, you're only looking for relative quantities of organisms. And you're only looking at organisms that you would remove with a swab. You're not looking at things living within the pores, within the hair follicles, in the crypts of the skin, etc. From my perspective, those are the most interesting organisms. But they're not accessible with this technique. You have to do a biopsy or something like that, right? And so they've got these hundreds of samples. They isolate DNA from each and every one and do PCR reactions and do high-throughput sequencing. Now, you can't sequence from all these different samples and do them independently. In high-throughput sequencing, you do them all at once, and you label the primers. And so the way this works is they're taking this chunk of the ribosomal RNA, and every PCR reaction, meaning every sample from every person on a single day, every single sample they take has a unique 5' primer when they do the PCR reactions. And that primer contains a barcode. All a barcode is a unique sequence that tells you, when you see that sequence at the beginning, you know, aha, this is person X on such and such a day from their armpit, left armpit. And so it's a way of determining what sample a given sequence comes from, because all these sequences come in one great big pile. That barcode also allows you to do error checking and quality control, looking at the shape of the peaks, and there's always a pair of nucleotides so you can tell how the shape differs, to make sure that the quality of the sequence is going to be good, to make sure it's not gibberish or anything else. They've chosen this region of the sequence because it's something that they can sequence all the way from end to end in a single reaction, and it contains some highly conserved sequences and some highly variable sequences so that they can get the maximum amount of phylogenetic information from this, even though you'd like to have the whole thing. But this gives you enough. they spend a lot of time figuring out how to display or rationalize the information. So what they end up with is 500,000 sequences. And you've got to do something with that to make sense of it. So one of the things they do is they create these bar charts. And so they drop them into these phyla, all the actinobacterial sequences, firmicutes, proteobacteria, etc. And this is from all the samples from all the time. And what you see is right off, the traditional gram-positive bacteria predominate in the human body. Lots of actinobacteria, lots of firmicutes. You also have a fair bit of proteobacteria and bacteroids. Some cyanobacteria. Wait a minute. Cyanobacteria? Where do you imagine this might come from? Yeah. Okay. I just remember what it said that. They take samples from your skin, right? You're covered in pollen. And the chloroplast of the pollen are cyanobacteria. You've got some fusobacteria here. But you'll notice that if you go to different locations in the body, those proportions vary quite a lot. And so in your mouth, there are a lot of firmicutes and proteobacteria. In your gut, it's a mixture of firmicutes and bacteroids. This is your skin, a lot of firmicutes, a lot of actinobacteria and firmicutes, etc. And you can go down the list. Pretty obviously, not really any cyanobacteria to speak of in your gut, because you can only breathe in and swallow so much pollen, right? Fusobacteria, that's predominantly in your mouth. Proteobacteria, also predominantly in your mouth, although found other places on your skin as well. This chart takes a little look at it, all right? It's pretty complicated. But it shows, I mean, this is a graph of the human body, obviously, and then samples from various parts and what are the different kinds of organisms they find there. This is divided much more finely than the previous one. The previous one had it all divided into phyla. This is more specific. And so, even down to genera in many cases, or at least families. And your eyes have to be better than mine to distinguish some of these colors. Let's see, where's Clostridium? Clostridium. So just for example, in your gut, there's a lot of Clostridia, a lot of bacteroids. And you can go through this then and look at the proportion of all the organisms here. One of the things you'll notice is that these have this giant gray area where there's no specification. A lot of times you get sequences that don't follow into these groups. That's where most of the diversity is, because they're individual sequences that don't drop into particular groups. I'll let you go through that for fun whenever you like. This one is maybe a little easier to understand. And so here they've divided this into different parts just as before, but a little more finely grained. And so if you look on the skin, for example, the predominant organisms are propionibacteria. These are actinobacteria, and they're anaerobes, that they are the predominant creature on your skin over most of your surface. This generally goes kind of from the top to the from the top of your head down. That's kind of the graph here, and you'll notice that the propionibacteria are predominant. The higher on your body you are, if you're standing up, the more propionibacteria there are. And the closer you get to the ground, the fewer there are. The sole of your feet, you're primarily things like staph. What are some other interesting ones in here? Micrococcus is a lot. Bionella, they don't have your mouth on them. Obviously, the genitals are a different case. I want to talk a little bit about this thing called unifrac. It's important. So if you want to compare these, for example, you have to have some quantitative measure. Take two populations and say, how similar are these two populations? This is important not just for comparing different proportions in different parts of your body, but different populations from the same person taken at two different times, or right or left, or diseased and healthy. How do you compare two different populations quantitatively? The measure for this is called unifrac. So what is unifrac? Unifrac stands for unique fraction. And so if you take all the sequences from two populations and you make a tree of them, what fraction, and so this is a tree, this is a superimposition of two identical trees. So if you were to take a sample from your palm one day and the next day, and if you got exactly the same population of sequences both times, you might get a tree that looks like this. And so the red might be day one, the blue sequence from day two. And you do the tree, and you get this thing, and you color code the branches based on whether that branch leads to a red or a blue sequence. These branches are purple because that branch leads to both a red and a blue sequence, right? And so if you add up the unique branch length, the branch that's only red or only blue, as a fraction of the total branch length of this tree, you get 0, right? Because none of these branches are either red or blue, and not purple. Here's two populations that are completely different. And so all the branches are either blue or red. And so the unique branch length is 1. It's 100% of the branches. Two real populations are going to be somewhere in between, right? So the unifrac for any two trees is going to be between 0 and 1. And the closer it is to 0, the more similar the populations are. And the closer it is to 1, the more different the populations are. Yeah?
It's called
Unifrac.
I guess unique fraction.
Unifrac. Unique fraction. And so you can take any two communities, generate these common trees, and calculate how similar the two populations are based on this method. And so at this point then, you can look, for example, right to left and say, okay, how similar are your hands to each other compared to maybe your armpits to each other? And determine that your hands are more different from each other, right and left, than your armpits, for example, because you can measure it. There's another method, which I'm not going to get into, because if you don't already know what it is, I can't explain it. It's called principal component analysis. Did any of you guys have the math to understand what principal component analysis is? Maybe a little bit. I'm not going to worry about it. It's a matrix method, and you essentially create then a plot that identifies the primary components of variation in a collection of numbers, and you can plot according to those primary axes. You end up with a graph that looks something like this, and the further apart the points are, the more different the populations are. The axes are arbitrary. They then go through, another one that they bring up here, I think, is this thing called an OTU, an operational taxonomic unit. So with this kind of ribosomal sequence analysis, we've already talked about how you never get down to the species level with this. But what do you get down to? What you get down to is this operational taxonomic unit. And this is an arbitrary cutoff. It has to do with how long your sequence is and how reliable your sequencing technology is, how well you can really distinguish two different sequences. And for the purposes here, and generally for this kind of analysis, any two sequences that are 97% or more identical are clumped together and called an operational taxonomic unit. That is, those two things are considered so close that they might as well be the same thing. Some people call this a molecular species. I worry a little bit about this because the word species has implications that we probably don't want to include, given that that would put all of the great apes in the same molecular species. So there's a lot of variation within one of these, but for the purposes of this, we can't resolve them. And so they took through all these samples, and they've got hundreds of bar graphs, and all of these are posted as accessory information on the website. You can take a look and find out something you're interested in. And so here's, for example, a plot of the oral cavity. You've got 6 people, and just the fraction of organisms that they have in them, shown as bar graphs. This is percent. And you can see there's actually quite a bit of variation. And so the Streptococcus is predominant in all of them, but not so much this person, who is male number 3, but quite a lot in female number 3. And this is variation from individual to individual. A lot of times these are pretty significant. I was struck right away by the amount of Neisseria in this guy's mouth. It turns out Neisseria is a common, not Neisseria gonorrhea, but other Neisseria species are common in the mouth. Bacteroids in the gut. Can you imagine what might affect the fraction of bacteroids in your gut? Your diet. Bacteroids are polysaccharide degraders. So the more starch you eat, the more bacteroids you have. And the more meat you eat, the more Clostridia you have. And so by looking at this, you can tell right off what a person has been eating. That's pretty scary, isn't it? You're also likely to see some parasites in here. And some that you can't really explain easily. This is the same data, though, based on date from a single individual. So you're looking now at variation from day to day, 1 day apart or 3 months apart. And what's really interesting is, for the most part, people are fairly consistent over time, at least over this time scale. People tend to be more consistent, obviously, between 1 day and 3 days. Yeah.
So when they took the gut sample, I assume they just took a...
It's a fecal sample, yeah.
So there's variation within the gut, essentially, right? So that's something that they found this out. This is mostly the large intestine.
Absolutely, absolutely. And it's not got anything to do with the biofilm that's on the large intestine. It stays put, for example. You're also talking about fairly heterogeneous samples sometimes. I mean, even your skin, right? I mean, there's different parts of your forearm that might be pretty significantly variable, like upper surface, lower surface, posterior. I mean, they're limited on how many samples they can take, obviously. What you'd like to do is take all different samples and look to see what kind of variation there is. Yeah. Absolutely. Absolutely. And so this is a principal component analysis, just to try to see whether you can tell one population from another. You might imagine, for example, that it would be a lot harder to tell your upper arm sample from your lower arm sample than it would be to tell your mouth from your bottom of your foot. And that's absolutely true. So this is a principal component analysis. The closer the dots are, the more similar the populations are. And it's pretty clear that you can tell the gut sample and the oral cavity from everything else. That's easy. The ears, pretty distinct, but not entirely. And then all the skin samples are kind of gamished together. You can also divide these by sex, by person, by day, instead of by habitat. And it's pretty clear that it's the habitat that's the principal thing that allows you to distinguish different populations. Certainly not day or person. By sex, a little bit. You might imagine these are certain parts of the body that allow this distinction. So where does variation occur? How do you look at these populations? Clearly, habitat is the biggest driver of difference between microbial populations. The people come next, and the time comes last. This is a phylogenetic tree of the populations, not individual sequences, but of the populations, and show how similar. This unifrac creates a number, right? And so you can create a distance matrix based on these numbers and generate a tree based on the population and variation within the population. And you see some interesting things. And so your right ear and your left ear are close to each other. Makes sense. Notice that for your hands, that's not the case. The palm and index finger of one hand is more similar to itself than it is to the homologous things on your other hand. Forearms, right and left, nostril, right and left, ear, right and left, armpit, right and left, sole of the foot, etc. It's your hands that are specific that way. Your hair is more closely related to your ear and forehead than it is to other parts of your body. The physical proximity, right? That makes some sense. And your gut is the most distinct one, and then things like your mouth and genitals are further out there. Some sites are more diverse than others. And so you might imagine that your gut has a very highly diverse population, but maybe, I don't know, the skin under your arms much less diverse. And that certainly is the case. And so they generate these things called rarefaction curves. And these are a little bit hard to understand. What they do is they take a subsample. So they've got hundreds of sequences from each of these samples. Pull all the similar samples together, so all the skin samples together, all the gut samples, all the oral cavity samples, and so they have these big giant piles of sequences. And then you fish out, for example, 50 sequences at random from each of these, and measure somehow the phylogenetic diversity of that subset. The easy way to do this is just tree them out and measure the total branch length of the tree. That gives you a measure of diversity. And then you take another sample, but of 75, I don't know what it is, maybe 150 sequences, then 300. And so you take increasingly large subsamples and plot the diversity, and it's a little bit like an enzyme curve, right? Theoretically, if you were to follow this out to an asymptote, that would tell you what the total diversity of that sample is. But in any case, the higher this is, this tells you that this sample is more diverse than this one, because there are more, for any particular sample size, you have more diversity in this one than any of the rest. So comparing these rarefaction curves gives you a measure of relative complexity. The scary part about these curves is that they don't plateau. No matter how many sequences you take, they never level off. What that means is that microbial diversity is a bottomless pit. But as long as you keep taking samples, you're going to get more sequences. But there is no end to microbial diversity. A little bit like looking at stars, right? So how does diversity compare? Here they're looking at phylogenetic diversity from different habitats. Okay, so female 1, palm and forehead. Clearly the palm is more diverse than the forehead. Some of these even run out of sequences. And so the palm of your hand has a much more diverse population than your forehead. And this is true for all of these individuals. Notice that the total amount of diversity differs from person to person. But always the palm is more diverse than the forehead. This is a graph that asks you how well can you tell two populations apart. how similar are different populations. And so here, for example, female 1, you can look at day 1 and day 2, they're related, and you can distinguish those from day 3 and day 4. This is obvious, right? If you take samples 1 day apart and then twice, 3 months apart, the day 1 sample is going to be most similar to the day 2 sample, and the day 3 sample will be most similar to the day 4 sample from the same person. But you can also tell the different people apart this way, and that variation is consistent across here. Telling them apart otherwise is tough. I urge you to take a look at all the supplemental data for this. All of this is here. Where is that? Here we go. This is those graphs. You can go through. Oh, I don't want to do that. There's a ton of data. What I want you to think about is what kind of questions you would ask. If you were going to do an experiment like this, what kind of questions would you ask? We're going to talk Friday about the human hand. What kind of questions would you ask of your hands? You wash them? Actually, you can tell that question. You can answer that question. By taking a swab, you can tell how long it's been since someone walked in. What other kind of questions? That's an interesting question. I don't know if they've answered that one. They can. One of the experiments they did is these guys work in a lab. It's a bunch of microbiologists, but it's also a bunch of computer people. They took swabs from the keys of all the computers in the lab, and they could match person per person whose computers they belong to, even for computers that hadn't been used in 6 months. They could identify which keys are the best for doing that. It turns out it's the space key. It's the most definitive information. They can tell whether you're right or left-handed by looking at the keyboard. What other kind of questions would you ask about it? Male and female is true. I mean, if you look at the data, it's a ratio of who is it? I can't remember who it is for male galactobacillus. It tells you right away, instantly, with absolute definition, whether a person is male or female. Yeah.
Can they tell like people who touch their face more?
They can tell people who put their fingers in their mouth. People like me who bite their nails. That shows up right away. Yeah.
You could ask where they live.
Yeah, so that's an interesting one. How does your work affect the microbial populations? It turns out it has a huge difference, makes a huge difference, but how that works is a little unclear. I used to work for an accountant, and I had my hands in gasoline all the time. Obviously, that has a huge effect on the microbial. Yeah. So people have looked a little bit at that. It turns out it's a lot more complicated than that. One of the experiments they did was they got permission at a marathon to take samples from people as they cross the finish line. And it turned out to be really easy to tell winners from losers in a race, based on the microbes on their hands. One thing they couldn't tell apart, by the way, was dollar bills. They collected dollar bills from all over, and it turns out that this is not funny. Dollar bills from a strip joint and dollar bills from a church could not be distinguished. So there's lots of those kind of questions that you can ask. What kind of food do you eat? What's your ethnicity? What's your age? All these are interesting questions that we often don't know the answer to. Yeah. Yes. Yeah. So that's an easy one. I think that one's been done. I think the answer is yes, but I don't remember for sure. Yeah, cats and dogs, that's really easy. I don't know about other kinds of pets. And it's not just your hands, by the way. They can tell from your from your mouth, whether you have a cat or dog.
Is that from like breathing in the pet
I have no idea. I mean, most of the time you have correlations, right? But you don't know what the causality is. So all you can do is look at that. One of the really interesting ones is if you take samples from people from all over the world, it's easy to tell people from, I don't know, Southeast Asia, from the U.S. The question then becomes why? Is that because of a difference in diet? Is that a difference in the work you do? Is it the environment? Is it your ethnicity? Is it the genetics of the people that are different? Detangling those things is really hard. And so it's also, that's a problem with interpreting this data, because you might look at a collection of people and say, well, these people have a different diet and we have this difference in microbes, therefore the change in diet causes that. But you may then realize that there's also a consistent ethnic difference or geographic location of people. And so detangling those things is not so easy. Notice in all that variation was within one population of people. Graduate student age people, these were all grad students in this lab. Graduate student, so young male and female people in Boulder, Colorado. Imagine how much variation there is amongst the human population generally. And all these people were healthy. So we're not looking at disease states or disturbed states or anything like that. You can imagine with this kind of variation that pinpointing a problematic microbial population is really hard. In some cases, it's really easy. So if you take a scraping of the teeth and you see a bunch of Prevotella, that's a danger sign. But it's not so easy. Normal is a pretty tough thing to identify when it comes to microbial population. All right? So, Friday, we're going to do the microbial handome. Oh, good question. I don't have them done yet. Those have been a lot harder to do than I thought, Greg, than I thought they would be. So it's taken me some time to get through them. What's that? Because the answers are not always what I expect, and so I have to look up stuff with a lot of the answers. So this is what we're going to talk about on Friday. Organisms of the hand. And we're going to prompt this by taking one of Rob's later papers that has to do specifically with the hand and looking at the populations on them. All right, good, thank you.